Okay, so today we are going to talk about the cell membrane. So our goals and objectives are to understand the fluid mosaic model of cell membranes, to describe the functions of phospholipids, proteins, and carbohydrates in membranes, to discuss membrane fluidity, to explain passive, tonicity, and osmosis and diffusion, and to understand active transport. So the plasma membrane and structure and function. So the plasma membrane has what's called selective permeability. So the polar phosphate head is hydrophilic, so it's going to be oriented towards the water, and our nonpolar lipids are hydrophobic, so they are oriented inwards. And this makes our phospholipid bilayer, which establishes our selective permeability. So this enables the cell to keep its cytoplasm separate from the environment surrounding the cell. Basically, the cell keeps its water and ions, everything else that it wants to keep inside. It does this through selective permeability, also while it keeps everything it doesn't want or what it considers the trash outside of the cell. The membrane also contains proteins, and they are a primary part of this membrane. They can be embedded, or they are embedded, and we have different types of proteins. We have integral proteins, and those are proteins that go all the way through this phospholipid bilayer, or they can be peripheral, meaning they can either be stuck on the top or the bottom. So here's an example of a peripheral protein right here. Also, you're going to find cholesterol in this membrane. You see a molecule of cholesterol right here. Now, you don't want too much cholesterol. However, it is vital that we have it for the fluidity of the cell membrane. We couldn't live without it. You're also going to see some glycolipids, which is a sugar attached to a fat. You can break that word down: glyco meaning sugar, lipids meaning fat. You're also going to see some glycoproteins like this right here, and again, glyco means sugar. Protein is, of course, a protein. So all these are found within our cell membrane. So phospholipids would also need to be tested for as an acid. So a phospholipid molecule is composed of a hydrophilic head and then two hydrophobic tails. And remember that these tails are long hydrocarbon chains, and they can either be saturated or unsaturated. So remember, saturated means that there are no carbon-carbon double bonds, so we have a greater saturation of hydrogens, and these molecules have straight tails, and they tend to sit on each other, and they're going to be solid at room temperature. Our unsaturated fatty acids, those are going to have a carbon-to-carbon double bonds. This will form like a kink in the tail, so our molecules can't actually become as compact against each other. So these types of fatty acids, they're going to be liquid at room temperature, and they're generally thought of as healthier fatty acids. So we have an example right here. You can see that this is an unsaturated fatty acid because we have that carbon-to-carbon double bond, as to where we have a saturated fatty acid over here because we have a lack of those double bonds. So there are proteins in our plasma membrane. There's going to be a hydrophobic region, and it's going to sit more out here with our polar heads, and then we're going to have a hydrophilic region, which is going to be in the middle of that phospholipid bilayer. So these proteins actually provide a pathway for the cell, or they can be used as some other function. So these phospholipids can actually move inside of the plasma membrane. If your diet is comprised more so of the healthy unsaturated fats, then your proteins are going to have a greater ability to float laterally from left to right. And this is happening from up to 10 to the 7th times per second. So as you can imagine, this is happening incredibly fast. Also, the phospholipid bilayer, they will actually flip once a month. So the entire membrane is actually going to turn over. So again, fluidity is affected by the types of hydrocarbon tails. So we can see here that we have more unsaturated hydrocarbon tails. So we see that little kink that I was talking about where we have that double bond. So we have more space, so we're going to have more lateral movement going on between these phospholipids, as to where we have a more viscous solution over here, meaning that's thicker. We have more saturated hydrocarbons, so there's no double bond, so all the tails are super straight and going to be really compact, so it's not going to be nearly as fluid. So this cholesterol, again, you don't want too much cholesterol, but we do need good cholesterol. In fact, it helps with the transport of hormones and other necessary items. Again, you can have too much though, which can lead to a less fluid membrane, and it can actually go on to clog your arteries and veins, which, as I'm sure you can imagine, is not a good thing. So again, we have membrane proteins. They can either be integral, meaning that they go the entire way through this phospholipid bilayer, or they can be peripheral, meaning that they just either sit on the top or the bottom. Again, we have some examples. Let's see, here's one right here. Also, you can see another one right here. So these integral proteins, again, they cross through this membrane completely. And remember that proteins have a certain shape, and that determines a certain function. So they have a certain job. And you can see here that we have a integral protein, and it's probably a tertiary protein because we can see the alpha helix. Then also you have, again, the peripheral proteins. And these will either be, again, stuck on the top or the bottom. So remember that proteins are extremely vital in our cell membranes. They serve several functions. So one function is the transport of ions. And remember that an ion is anything on the periodic table that has a charge. It can be either positive or negative. And this type of transport often requires energy. So we see here that ATP is helping with the transport of this molecule. There's also channel proteins, which this looks like it would be a passive diffusion. There's no energy needed, but it's going through a channel protein. Then we also have enzymatic activity, and that just helps lower the energy needed for an activation. Then we also have signal transduction, and that's when these proteins will act as a receptor. Something will bind to it, and then it will let the cell know that some kind of work needs to be done. We also have cell-to-cell recognition. So this lets a neighboring cell know that a certain cell is part of you, and not to release white blood cells to either come and try and fight it or possibly kill it. So this is a big part of our immune system. Of course, you don't want your body attacking itself. There are a couple of diseases where this is an issue. We also have intracellular joining, and this is where our intercellular communication is going on. So these cells are affixed to one another, so this is allowing them to communicate. And then we also have the attachment to the cytoskeleton and the extracellular matrix, which you can see here. So carbohydrates can play a large role in the cell-to-cell recognition. So that's the cell's ability to distinguish one type of neighboring cell from another. So a good example would be the determination of your blood type. So on every red blood cell in your body, there is a glycoprotein that determines your blood type. So if you have type A blood, you wouldn't want to receive type B blood from another individual. The cell of the donor would not be recognized by the cells of the receiver. So this would cause blood clotting. So plasma membrane structure has a selective permeability, and this controls the cell's exchange of materials with its surroundings. So the cell only wants to let certain things into the cell, and remember that the outside of the cell is what's going to support the function of the inside of the cell. So it wants some things, but it doesn't want everything. So this depends on the interaction of a molecule and the hydrophobic core. We have what's called aquaporins, and these are channel proteins that will move 3 billion water molecules per second into the cell. Now, despite this, it's still difficult to get water in and out of the cell. And even better, this becomes more difficult as we age. So as we get older, we are more likely to become dehydrated. And this is why it's very important to make sure that our seniors get plenty of water. So the permeability of this phospholipid bilayer is determined either by the hydrophobic molecules, which you should remember are our nonpolar fatty acids that are oriented inwards. So these particular molecules, like other lipid solid molecules, but there are some things that can pass through the membrane rapidly, and that would be other hydrocarbons, carbon dioxide, and oxygen. Polar molecules, these don't want to cross the membrane quite as rapidly. So what can you do to tell if something's polar? Well, you can look and see if it's dissolvable in water. Check and see if it has any unpaired electrons or if it's not symmetrical. These are usually your polar molecules. So there are two different types of transport proteins. Now, transport proteins allow the passage of a hydrophilic substance across the membrane. So again, we have two types. We have our channel proteins, and one example of that would be our aquaporin, which again helps get water into the cell. And then we have our carrier proteins. So we can see here we have a channel protein, and it literally just looks like an open channel. Our carrier proteins kind of looks like it's kind of like cradling this little molecule right here, and then it spits it out to the other side. Okay, so we have a couple of different types of transport. We have passive transport, and passive means that there's no energy required. So this is the diffusion of a substance across a membrane. Again, no energy is used, and we have a couple of different types. There's just diffusion, there's osmosis, and then we have facilitated diffusion. Then we have active transport, and active transport, just think if something is active, then you're using energy. So energy is required to move these substances across the cell membrane. So passive diffusion or passive transport or just diffusion is the tendency for molecules of any substance to spread out evenly into an available space. So this is the movement of substances from a high concentration to a low concentration. So how does it flow? High to low. So this is random movement, and it's driven by kinetic energy because we have constant movement and these molecules are constantly bouncing off each other, driving that movement. Now this is going to happen until we reach equilibrium, which you can see that right here. It looks like there's about an even amount of the molecules on each side of this membrane. You may still have a little bit of movement back and forth from high to low, but it's pretty much going to remain at equilibrium at this point. So substances diffuse down their concentration gradient from a higher to lower concentration. So again, how does it flow? High to low. And a concentration gradient is created when there are more ions of a substance on one side of a membrane than another side. Remember, everything is seeking equilibrium, so it's going to go from high to low. So osmosis is a special type of diffusion. It's the movement of water across a semi-permeable membrane. So osmosis, just think the diffusion of water. So it's also going to depend on solute concentration. And then we have what's called osmoregulation, and that's the control of water balance to maintain an internal environment. So tonicity is the ability of a solution to cause a cell to either gain or lose water. Now you're going to have a greater impact of tonicity on cells that don't have cell walls. So think of an egg that doesn't have its shell. So if you were to submerge that egg in a cup of water, well, it doesn't have that really hard, rigid cell wall or eggshell on it to prevent the movement of water. So it's going to more easily cross through this egg that doesn't have its shell. So we have three different types of tonicity. We have hypertonic, isotonic, and hypotonic. So let's start with hypertonic. So something is considered hypertonic if it has a greater concentration of solute outside versus that of the inside. So Let's pretend that you're lost at sea. It's been several days since you've had anything to drink and you're incredibly dehydrated. So you think that, okay, I'll take a sip of the water. So you take a sip of the water. Well, the concentration of salt is much greater now outside of your cells versus the environment inside of your cells. So that concentration of salt is much greater outside, so water will rapidly rush out of your cells in an attempt to even this out or find an equilibrium. So those cells are going to shrink. So we see here water is rushing out. Now let's pretend on another hand that you have a friend who has dared you to drink a massive amount of water in a very short period of time. So you've now you've taken in quite a bit of water. So now your concentration gradient is greater inside of the cell compared to that of the outside of the cell. So now water will will rapidly rush into your cell, so much so that the cell could possibly expand and go through what's called cytolysis, which is cell bursting. Both of these are incredibly painful, which is why we want to be here at this isotonic solution where everything is at equilibrium. So cell walls can help maintain water balance in plants and fungi. So we have plant cells in the three different types of tonicities. So this plant cell has a greater concentration of solute inside the cell. So water is going to rush into that cell. It's going to make the central vacuole absorb more water. That's going to press the cell membrane against our cell wall. It'll press the cell membrane against our cell wall, which it's pretty rigid, so the cell membrane will not push past that cell wall, but it will make our cells swell. And this creates what's called turgor pressure, which is what allows plants to stand the way they do against gravity. So an example of this would be if you went on vacation for a couple of days and you forgot to water your plant, I mean. If that plant was a little limp when you came back, it's a little dehydrated, but you can give it some water, then that water is going to rush in, and then after a couple of days it should be normal. So again, that's how we get turgor pressure. Now over here we have a plant cell that is in a hypertonic environment, meaning that the concentration is greater outside of the cell, so water is going to rush out, and that will actually cause the plasma membrane to pull away from the cell wall, except at these plasmodesmatas right here. So the cell membrane, or the cell itself, is going to shrink, and that's called plasmolysis. And then we have a flaccid cell, and that is just when the cell has the same concentration of water and of solute, both inside and outside the cell. So here we can see what animal cells would look like in hypotonic solutions, isotonic solutions, and hypertonic solutions versus plant cells. So facilitated diffusion is another type of passive transport, so no energy is required. So this will use transport proteins to speed up the movement of molecules across the plasma membrane down their concentration gradient. So the transport proteins, we have two types. We have channels, and then we have carrier proteins. And we can see here the different types of passive transport. We have diffusion, and then we have facilitated diffusion. Either through channel proteins or carrier proteins. And don't forget there's also osmosis, but that's going to be our water diffusion. And then we have active transport over here, which does require energy, and this is done via ATP, and that's going to push something down the concentration gradient. So active transport requires ATP, or energy via ATP, and it's going to move a solute against a concentration gradient. So it's making it go a way that it wouldn't naturally go. So that's why we need that extra help. So an example of this is a sodium-potassium pump. So one ATP is used, and that will push 3 sodium cations outside of the cell in exchange for 2 potassium ions being pumped into a cell. So this sodium-potassium pump is used several places throughout your body. It's very important in neural transmission. It helps with kidney filtration. It's also very important in cardiac function. So membrane potential, you have electrical potential energy due to a separation of opposite charges. So this is going to be considered a chemical difference. So we'll see this where the cytoplasm is going to have a slightly more negative charge compared to the extracellular fluid outside the cell, which is going to have more of a positive charge. So an electrochemical gradient is caused by the concentration electrical gradient of ions across a membrane. We see here that we have a more positive charge in this extracellular fluid and a more negative charge in the cytoplasm of the cell. So we're going to have to use ATP to actively push this positive proton through this proton pump outside to the extracellular fluid. Normally anything positive is going to have a greater affinity for something negative. That's why we need that ATP to push this against that concentration gradient. So an electrogenic pump will transport proteins, and it will generate a voltage gradient across this membrane. So we see here we have that electrochemical gradient created by the difference of charges on each side of the membrane. Again, so we have in humans, we have sodium potassium pumps, and that's actually in animals, not just humans. And then in plants and bacteria and fungi, we have this proton pump that you see here. Again, both of those are a type of active transport, so they're going to require energy in the form of ATP. So co-transport is another type of active transport. And this is of a specific solute that will indirectly drive the passive transport of another solute. So the active transport of this proton will passively help in the transport of another solute back into the cell. So the bulk transport of large molecules, there are a couple of different terms. We have exocytosis, and this is when the vesicles that are coming from the Golgi apparatus will actually transport materials that no longer need to be in the cell. It will help it exit the cell. And this is done through exocytosis. So exo, think exit. So it's going to release their contents out of the cell. Then we have endocytosis, and this is how we take in those macromolecules. So there are a couple different types of endocytosis. The first one should sound familiar to you. We have phagocytosis, which is cell eating, and then we have penocytosis, which is cell drinking. And then we have what's called receptor-mediated cytosis, endocytosis, which is whenever a molecule will have some receptors on its cell membrane. It'll actually come over to this other cell that will have receptors as well. They'll match up, and a vesicle will form around the molecule, and it'll actually come into the cell. So let's go over the tonicities. So we have an artificial cell consisting of an aqueous solution enclosed in a selectively permeable membrane. And it has just been immersed in a beaker containing a different solution. The membrane is permeable to water and the simple sugars glucose and fructose, but it's completely impermeable to the disaccharide sucrose. So which solute will exhibit a net diffusion into the cell? So first things first, let's go ahead and cross off our fructose, or our sucrose. Alright, so now we are going to look at the amounts of glucose and fructose, both inside and outside of the cell. And let's get rid of that sucrose. Alright, so we have 0.01 moles of glucose and 0.01 moles of fructose outside of our cell, and we have 0.02 moles of glucose inside of our cell. So let's first look at the glucose. We see that we have 0.01 moles more of glucose outside the cell than we do in the environment. So that glucose is going to leave the cell and go into our outside environment. Now for the fructose, we have more fructose outside of the cell than we do inside the cell. We have 0.01 moles of fructose as to where we have no fructose inside the cell. So about 0.05 moles of fructose are going to readily enter into our cell. So which solution is hypertonic to the other? So in this case, let's look at the sucrose as well. So we have 0.05 moles of a sugar inside of our cell. So let's say we have 100% water. We're going to subtract that 0.05 moles. So inside of our cell is going to be 99.05 or 95% water. And outside of our cell, we're going to add these up. We have 0.03 moles of sugar outside of our cell. So that's going to leave us with 99.97% of water. So the amount of water outside of our cell is greater, so that water is going to rush into the cell, and that makes our cell hypertonic to our hypotonic outside environment. So I just answered your last question. Your water should be moving into the cell because the concentration gradient is higher in the cell. So that water is going to try to reach equilibrium by entering the cell. So let's do a quick recap. You should have a better understanding of the fluid mosaic model of the cell membrane. You should be able to describe the functions of phospholipids, proteins, and carbohydrates in membranes. You should be able to discuss membrane fluidity. You need to be able to explain passive diffusion, tonicity and osmosis, and diffusion. And you need to be able to understand active transport.